Hey guys, welcome to the video on chapter 12 section 3. I have 3 goals for you guys today in this video. First off, I want you guys to be able to explain how DNA is used to create proteins. Secondly, I want you guys to be able to take a strand of DNA and turn it into a protein. And finally, I want you guys to be able to outline the process of translation and transcription. So, I want you guys to focus on this essential question as we're coming in. How does DNA become a protein? Well, to answer that question, we have to go into what's called the central dogma of biology. And the central dogma of biology is a really fancy term. It sounds really impressive, but really all it means is 3 things. It means that because DNA starts off first, you start off with DNA, that DNA then goes to RNA, which then turns into a protein. So it's a 3-step process. So you start off with your DNA, you then turn it into what's called messenger RNA, which that gets turned into proteins. And that's all the central dogma really talks about, is that DNA, RNA, protein. So I guess we're forced to ask a question to start off here, and that is, what is RNA? RNA is very similar to DNA, except it has a couple of small differences. First off, it has uracil rather than thymine. So in DNA, what you guys know is that if I had like A, and then G, well then the other strand would be T, T, C. However, in RNA, what happens is this becomes U, U, C, because the thymine gets replaced with uracil instead. That's the big difference that you guys will have to know, and it'll actually make a difference for you guys as you work through your problems. There is one other difference as well, is that there is a sugar molecule that's part of the RNA molecule that's different from DNA. But the big one here, guys, is to understand that uracil is the big change. So rather than having thymine, you now have uracil. Okay, so RNA comes in 3 types. There is mRNA, which its job is basically to carry information from the DNA in the nucleus to the ribosomes. That's really all the mRNA does, and that'll make a little more sense here in a little bit as I explain more about what mRNA does and how it works. Now, rRNA is a fancy way of saying ribosomal RNA, and basically that just looks like this right here. It's just a molecule, it's a combination of RNA molecules that form something we call a ribosome. Now, if you remember from the cell biology part, ribosomes are the organelle that makes proteins. So now you know that ribosomes are actually made up of RNA. The final one is called a tRNA, and a tRNA basically transports amino acids around the body. So amino acids, again, this might be a key term you might remember from last year, but amino acids are the monomers of proteins. So remember that when you see the word amino acid, we're just talking about the monomer of a protein. And tRNA's job is to carry those around. Now, that's the basics here, guys. Like, gives you an idea about all the key terms you're going to be seeing here soon. The rest of this, guys, is process-based. It's understanding what's going on. Well, to understand that, you have to understand the very first thing, and that's what's called transcription. Transcription is just the taking of DNA and turning it into messenger RNA. So if you see down here on this chart below, what we see here is that an enzyme called RNA polymerase is attaching itself to the DNA, and it's taking the DNA molecules on this side, and it's attaching mRNA to the top part. And what happens is that essentially gives you one big strand of mRNA. And that mRNA is going to come into play here in a little bit. So it basically uses the DNA and turns that DNA into mRNA. So here's an example right here. This here is a sequence of DNA. So we have A, A, T, A, C, A, T, T, C, A, T, T. And here's the 5' direction, here's 3' direction. Now, what's going to happen is that an RNA, when RNA polymerase is going to come onto this DNA molecule, and it's going to create an mRNA molecule with this template right here. So what's going to happen is that an mRNA molecule will be created, and it's going to create, well, let's take a look. Well, A bonds with T, usually, but we're dealing with RNA here, so that means that's going to be a U. Well, A goes to U. Well, here is a T. Well, T goes to A goes to U. Well, C goes with G. And we'll just keep on going. So we have A to U, A to U, A to U. T is going to go to A. This will be a G. This, because we have C goes to G. I actually screwed that part here. That's a G. There we go. And this here will be, again, another U. This here will be a G, and this here a G. So now, it creates it just the same way it happens in DNA replication. So this will be a 3' end, and this will be a 5' end. And this ends up being a molecule of mRNA. So the DNA here, the DNA is used to create a strand of mRNA. And this is what that would look like. Here is a sample strand that I've created. So what happens next? DNA has been turned into mRNA. Well, what's the next step? Well, the next step is that the mRNA has to be fixed. We don't really know why this happens in biology yet, but there's two things that occur. We have things called exons and introns. What happens is this, is that an mRNA molecule, so this big here is an mRNA. This big strand here is one big mRNA molecule. Well, what happens is that these sections we call exons, these get cut out of the mRNA molecule and get put into another mRNA molecule. And these parts called introns, which are the parts right here, these parts get removed by enzymes, leaving you with just the exons. So the introns are the RNA that is removed from the final mRNA molecule. So this here would be my final mRNA molecule. And this here is my initial mRNA molecule. The exons become part of the final molecule, while the introns are deleted and removed. So, once you've created your mRNA, the very next thing that happens is that the exons and the introns are separated from each other, and we create a final mRNA molecule that's made of just exons. Now, once you have that, what's next? Well, the next part is we call translation. Translation takes this code of mRNA and turns it into a protein. Now here, guys, I've shown you this before, but I didn't show you how to use it. This here, guys, is the code of life. This here tells you how mRNA is turned into amino acids. Now, you guys already know this here is the mRNA molecule. Now what happens is this. Every 3 bases equals 1 amino acid. So every 3 of these letters equals one of the amino acids on this table here at the right. 2 or more amino acids combined together equals a protein. So if you have more than 1 amino acid that's combined together, you have a protein. So let's go ahead and take a look here. Now, there's a trick here, guys, and I put it in here to see how it works. Now, you might sit there and think, well, obviously, my very first amino acid is going to be UUA. Well, let's look over here real quick. Well, here's U, and I want UUA. Well, that's right here. That's an That is an amino acid called leucine. So I'll put a L here for leucine. And you might think that makes sense. It looks good, doesn't it? Because I have 3 bases. I have the code here that says leucine, so that means my first amino acid is leucine. However, the code's a little more complicated than that. What really happens is that you can't actually code for anything until you've hit the start button. And you're probably like, start button? What are you talking about? If you look right here, there is an amino acid called methionine. It's called the start codon. What that means is that this will only start making amino acids once it's encountered the letters A, U, G together. So the UAA happened before we had any kind of a signal to start. So this wouldn't work. So the 2 U's here don't turn into an amino acid. However, if you look right here, you'll see you have AUG. Well, AUG is methionine, which means that that's going to be your start. So I'll put an M here. Oopsies. I'll put an M here, and I'll put in parentheses here start. Once AUG is discovered, then it starts coding every 3 letters. So let's take a quick look. So my first one's methylene. Now UUU, let's look it up. UUU is right here, and that stands for phenolphthalein. So I'll put a PH right here for phenolphthalein. Well, my next one is going to be AAG. So let's take a look. Here's A, here's AA, well, here's AAG. The next one is lysine, so I'll put a LY for lysine. Now, the next one I have is UAA. Well, let's take a quick look. It's around here. Here's U here. Here's A. Okay, UAA. Oh, UAA stands for stop. That means that you no longer code anymore. So my protein is a combination of 3 amino acids. It's methionine, phenolphthalein, and lysine. And bam, I have now just turned a sequence of mRNA into a series of amino acids. And this here, guys, would be a 3 amino acid protein. Amino acid protein. And there you go. That's how you can take mRNA and turn it into a protein. Now, I didn't bother going into detail about how this happens. So here's what happens, but how does this happen? Well, that happens through a process right here. So here's your ribosome. So this here is the ribosome. So I'll put R-I-B-O for ribosome. And what happens here is this, is that this green line right here is your mRNA. And these right here, if you don't remember from the first slide before, these here are tRNA molecules. What happens is that this tRNA has a series of bonds to them that will bind with AUG. What ends up happening then is that this tRNA molecule is going to carry the amino acid methylphenylene. So I'll just put meth. Now that'll be this circle right here. Then this tRNA molecule goes with AAA. So that means that you'd have to see a UUU in order for it to work. So this here would attach, if we go back, so UUU, we go back to our code beforehand, well, that would be phenolphthalein. So that means that this here is going to attach phenolphthalein. This right here, the tRNA molecule here, well, let's see. It's going to be, the codon here is going to be ACG. Well, let's go take a look back here. So ACG. That means that tRNA molecule is carrying an enzyme called threonine. I have no idea how to pronounce that. So that means the next enzyme here to attach will be that enzyme threonine. So that's how this works, guys. Now, there's some key terms here you're going to have to know. First off is the word codon, and secondly is the word anticodon. The codon is the part of the RNA molecule that has the 3 letters that tell you what amino acid you want. The anticodon is the part on the tRNA molecule it's going to attach to the codon here. And that's how this works. So basically what happens is that a tRNA molecule is going to bring in an amino acid that's going to attach to another amino acid, and eventually you're going to create one big protein chain full of amino acids that are attached together, making a protein. And that's how that works. Now, a couple of things about ribosomes. Ribosomes are made up of 2 parts. There's the 60S part and the 40S part. The big part here is the 60S part, and the small part here is the 40S part. The S just stands for a subunit, so it just means 60 subunits. Now you don't have to really know what that means, just know that S is a way of measuring how big a ribosome is. And every ribosome is made up of 2 parts. The top part right here is going to hold the tRNA, and these sites right here are where the tRNA molecules are going to go. The 40S part right here, its job is to hold the mRNA molecule. Now, ribosomes are made up of basically just 2 things. They're made up of RNA and proteins combined. Now remember, when we talk about ribosomal RNA, we call it rRNA. And that's pretty much it, guys. One last idea. So I have all this information here. What's the connection? So you guys know that there are genes, you know there's alleles, but you don't quite know how that all works with this code. Well, what happens is this. Every single gene codes for one enzyme. So for example, the gene for sickle cell is just the enzyme for sickle cell. So when we talk about a person who has sickle cell disease, all we're really saying is that they're making a protein that gives them sickle cell disease. So what's the connection here? How does this work? Well, it works like this. Diseases in genes. Recessive diseases, guys, are usually just broken enzymes. They're proteins that don't work. For example, let's take hemophilia. Hemophilia is a recessive trait. That means you have to have small r in order to get it. What this means is that when we talk about these small r's, big R's, all that for alleles, all we're really saying is that the R stands for an enzyme. In this case, since hemophilia is a disease and it hurts you, the small r is a broken enzyme. Which is why, if you have a big R, the dominant trait, if you have the big R, you're okay, because the big R is a working enzyme. That's the connection, guys. A dominant trait, so I'll put DOM for dominant, just means that you have a working enzyme. ENZYME. A recessive trait usually means the enzyme is broken. That's the connection, guys. That's how this stuff works. Now, it's not always that simple. There's more to it, and we'll go into that later on. But for now, guys, it's good to understand that a dominant trait, the dominant allele, usually is just the working allele. It's the one that actually works. Whereas the small trait, the small letter, and the recessive trait is usually an enzyme that's broken. And that's it, guys. Here are the key terms in the chapter. Here is something I'd like to see answered in your Cornell notes. It's essentially the essential question from the very beginning. So you're going to want to try to answer this question, and I hope this is helpful, guys, and enjoy the activities.